So as Mark mentioned, we've just started an interdisciplinary academic effort with the goal of trying to pick up some of the slack that's being left by an industry shift away from early lead discovery. And so that also serves as an advertisement for this afternoon's session to talk about how the economics and landscape are changing in drug discovery. So this involves, in addition to myself, a number of investigators from across the Harvard landscape. Again, our goal is to take an academic approach, which is different from an industrial top-down approach to identifying lead compounds for treating specifically MRSA and VRE. We have three drug discovery arms. One is Suzanne Walker, headed by Suzanne Walker at the med school. Another one headed by Lefteris Mylanakis and Fred Alsobel at Mass General. Another one headed by Roberto Coulter and Rich Losick at the med school and at the Faculty of Arts and Sciences. Then we have two validation and resistance elements, one headed up by David Hooper at Mass General and one by myself. And then advising us as we go are Chris Walsh, who has considerable experience at the interface of academics and pharma, Nathaniel Gray, who's a medicinal chemist, Jared Silverman at Cubist, and Steve Projan, who is at Novartis and now is at MedImmune. So just very briefly, what Suzanne Walker's doing is she's engineering high-throughput screens to identify inhibitors of directed targets. Her first effort was to identify a conditional lethal target of the wall-teichoic acid biosynthesis pathway. Then Fred Alsobel and Lefteris Mylanakis are using a really novel screen. They're using C. elegans, and they have the robotics to add exactly 15 C. elegans to a well of a 384 well plate. And then they'll add 70 microliters of medium that has either Enterococcus or staph in it. And over the course of 3 or 4 days, the bacteria will kill the nematode. And they have an optical device that reads this. So when a nematode dies, it goes stiff and settles to the bottom of the well. When they're alive, they're squiggling around, and that scatters light differently. So they can read these 384 well plates in real time. And so they use these plates just like any other plates in a high-throughput screening assay to sample large compound libraries. The interesting thing is about a third of the compounds that save the life of the worm are traditional antibiotic-like compounds that when you retest them have an appreciable MIC for the microbe. 2/3 of the things that save the life of the worm, however, don't have MICs, which is really interesting. About half of those seem to activate the immune system, and then the other half are just complete mystery compounds. So they're discovering things that save the life of the worm, which is really an outcome that we're interested in. But by nature, they're also biocompatible, because if the compound killed the worm directly, if the compound was toxic, it would fall out of the screen. So these are screened for biocompatibility, and they're screened for basically the end result. So that's really an interesting and a novel, and that's the kind of thing you can do in academics, for example, that's rarely done in industry. Then Roberto Coulter and Rich Losick are looking at biofilm disaggregation principally. They noticed that with time biofilms disaggregate, and they selected for some mutants, and they found some pathways that are essential for disaggregation of biofilms, and they're targeting that. David Hooper is focusing on efflux systems in MRSA strains and assessing the extent to which lead compounds coming out of the discovery elements are subject to the known efflux pathways. We're working on resistance, movement of resistances between staph and Enterococcus, for example. And we're also examining lead compounds in mammalian model infections. So that's more or less how it works. We started off 2 years ago as a pilot project. In that pilot phase, Suzanne came across a very nice looking lead compound that inhibits the tar-GH step in wall-teichoic acid biosynthesis, made a second generation molecule based on some structure-activity relationships that had MICs in the 1 microgram per mil range, and was equally effective for MRSA as well as sensitive strains. The nematodes study has turned up about 30 interesting lead compounds. About 10 of those have MICs for the bacteria. About 20 of those are sort of the different acting compounds. And we've just launched the real phase of this for 5 years, supported by NIAID. So we look forward to seeing if academics can take some new approaches to drug discovery to help fill the vacuum at the very front end of the discovery pipeline. And we look forward to interfacing with all the other ongoing efforts in the Boston area, like Mark's and the prudent use group at Tufts and the many other efforts. So that's what we're thinking. So with that bit of introduction about that effort, let me start the discussion session. So before we ask for questions from the audience, let me take the organizer's prerogative, and in the meantime, you can think about your questions. But let me, as chair, ask, Susan raised the point about the details. Do we need to know what strain we're talking about, what lineage we're talking about when we do these studies? And even more so, one of the fundamental questions I had is, when we sample the nares, we're sampling maybe 1 square centimeter or 2 square centimeters of epithelial surface. And we find about 25 or 30% positivity in staph carriage rate based on that 1 or 2 square centimeters. Knowing that the epithelial surface of a human is on the order of square meters, if 25% are positive in that, and as we heard from Michael, maybe the nares isn't always the best place to sample, what is the real positive rate for the body, first of all? And secondly, if in terms of SSA and MRSA co-carriage, our sensitivity to detect that is maybe only 1 in 100, at most 1 in 1,000. But if we were to take a deep metagenomic search into the body, what are the chances that we would find an MRSA in that body that would then bloom out on antibiotic treatment? So 2 questions. How much do we need to know about the details to really know to get to the bottom of the problem? And secondly, are we sampling well enough? Anybody have a comment or thoughts for that? Michael?
Well, I completely agree with you that we do need to know the details. We do need to know the strain types. In the study that I cited up there, where we tested 1,100 people, we actually did genotype all of those strains that we derived from those people from all 3 sites. And we did find a number of people who had co-carriage. I can't cite the percentage right now, but it's higher than 2%. Strains act very differently. So, for example, USA 300 is much more likely to be found in a non-nasal carriage site than other strains, than healthcare-associated MRSA strains in our study. One important variable that really affects the literature as well is if people sample, do these swab cultures in very different ways. Some people do overnight enrichment broth culture, which we did, which definitely enhances the sensitivity of the assays. So I completely agree that the strain type, it's really essential. These bugs act different ways. They cause different types of infection. They colonize different groups of people. They are transmitted in different environments. So I mean, to me, I think without knowing the strain type, there's a lot you don't know.
Okay, thanks, Michael. Susan, what are your thoughts on this?
I think we need more epidemiologic data that shows what the correlates are really for infection. And I think that in some cases it might matter what the strain is, in many cases it may not. And there are other issues that are in play, including the burden of carriage, not just whether it's carried in certain sites. There's evidence that if you don't clear the nose, you really can't clear other places for certain types of healthcare-associated infections. There's good evidence that has not yet been published by the CDC, but that the strain actually doesn't change the distribution of nosocomial infections that occur in the healthcare setting, even though there is quite different in the community. So I think that what we need to do is, there's a lot of detail out there as we go into genomics and do high-throughput sequencing. You can learn a great deal, but I think we always have to step back and make sure there is some valuable epidemiologic and clinical correlate that makes a difference for why we might do these types of tests. And in the end, it may just be that certain types of host factors, diabetes, wounds, may really drive and be the dominant reason why you need protection in certain hosts, versus smaller, less predictive characteristics that might relate to location or genetics, or might be completely the opposite, but I suspect that there's only a handful that really make a huge difference, and we should really target our efforts in that domain.
Thanks. Those are good points, and certainly nasal carriage probably is an indicator of high-level carriage, at the very least. I think that's probably true. Questions from the audience? Mark?
Comment and a question. One is, in thinking about comparing some of these presentations today, it was really interesting from Ed to see that Ankara and Istanbul are essentially the same place from the perspective of Staph aureus, and Izmir is separate, whereas in Orange County and maybe in the UK and the Netherlands, the spatial scale of sharing appears to be possibly smaller. And it'd be interesting to know whether that's because the technologies are different, a genetic versus a sort of prevalence-based estimate, or whether it's that maybe there's a lot more patient sharing between Istanbul and Ankara than there would be between places of that distance in another setting. So just trying to combine some of these measures. It's, again, this marriage of genomics and epidemiology would be interesting. The question is for Chibe. You emphasized the network effects on the risk of MRSA, and then you pointed out that when the government said you got to get your MRSA down, they did it, which I imagine maybe was not mainly through network interventions, but rather through same hospital interventions. So the three possibilities are: A, it was through network interventions; B, they were cooking the numbers, which can happen in some places, and you seem to be maybe suggesting, although I don't know, or C, that in fact the internal dynamics of the hospital really do matter. And I'm curious what several people think.
Well, I think that if all hospitals take their own measures on a single hospital level, you will still see an effect. Although it might have been more efficient to create a sort of a regional or a national scheme in which all hospitals sort of combine their efforts and find more efficient ways of dealing with it. But still, if you get all hospitals on a single level to try and reduce their rates, it will inevitably have its effect on the total rate in the entire country. Yes.
I think some of the issues that might be involved is what's being measured right now is invasive disease. And yet, and as invasive disease comes down, because we protect devices and other types of things that put people at risk, prevalence continues to rise. And so at some point, there is a real balance between whether you do anything for the person who walks in with it, where the hospitals could care less because it's not their fault, versus what you're going to do when it reaches some critical threshold, and then the risk becomes quite substantial, whether it's community-based or not. And then I think that's part of the real push in the United States to look at post-discharge events that circle back to the hospitals and hold hospitals accountable.
Regarding the Turkish data, in your point about the hospitals in Istanbul and Ankara being mixed, I agree, it is interesting on the face of it. It is a contrast to some of the other data we've seen today. However, there are two caveats. One is that I have no data at all on the amount of patient movement in that system. And the other is that the number of isolates are actually quite small. So although it's quite telling, and we've done some more sophisticated analysis saying that they really are quite mixed up, I would just hold fire a little bit on inferring too much from that, because we're comparing very different systems, and this is one particular clone, remember, that we're talking about. It's a very, very high resolution data.
I just follow that up just briefly. I noticed, Chiba, on your data, it looked like Cambridge was an outlier in terms of not coming down with respect to MRSA rates. It was, and that includes Sanger, right? Is Ed behind that, or what's Ed Brooks not washing their hands, or what?
I don't know what's going on there. It's no, I don't know why some clusters of hospitals stay high and others decline. I don't know.
Yeah, I was just curious.
Yeah.
Had they not implemented things, or is it because it's such an academic hospital, or
Well,
both?
all these groups of hospitals have academic centers within them. So it's not that it's the academic center, so
Okay.
Addenbrooke's, but no. So Oxford, the cluster of Oxford, does decline, and they've got a pretty big hospital as well.
All right, very good. Yes.
Well, I guess my question is related to what you're saying, and it's for Chiba and Susan, I think. So if you have all these predictions, I guess you can also find the hospitals or nursing homes that are doing particularly well compared to what you would expect. And could you learn from those places? Right, so the ones that have a lot of or low MRSA rates, even though you predict that they should have high rates.
Yes, you can find them. There are hospitals that have very low rates. But then again, there might be other factors playing in there as well. You need to take more data into account. Patient population, for instance, may matter. Antibiotic use may differ between regions, things like that. But you can find them, yes. I don't know.
One of the things that we're looking at is across the nursing homes in Orange County, what is it about nursing homes that enable them to import a certain amount and maintain it versus those that import a certain amount and then it looks like there's some explosion of transmission. And the nursing homes are perhaps in some ways even more difficult to study than the hospitals because there is a great encouragement for a home environment. There's a real desire to mix and mingle, which is really different than most hospitalized patients don't go next door and hang out with the next patient. So we have started to look at things like environmental sampling for having contamination in public areas where they share meals and so forth, and have found a correlation between those that are able to maintain things and keep things at bay versus those that look like they have transmission risks. So I think they're hard studies to do, as you can imagine. They're not, you don't get the value of all the people. You have to take each center or each nursing home or each hospital by itself and gather a large number of them to make inferences. But I think it is important to do that. And I think that the goal for at least the US in identifying outliers is to learn from the best and to avoid what the worst are doing.
One thing our modeling is also emphasizing is that it's a dynamic system. So if particular hospitals are doing better, it may be something within the hospital, but also you have the connections to other hospitals matters, and the infection control within those hospitals that the hospital is connected to matters as well. So we're trying to bring the focus from just single hospitals to the whole connected system.
Yes.
So question for Ed. So you constructed phylogenies using only the core sequences that are present in all isolates. Can you go and map back onto those phylogenies all the non-core sequences, and are they doing interesting things? Sort of related question. You also mentioned that virulence is apparently increasing. Can you see what is behind that using the molecular data?
Okay, so the first part of the question is really, really interesting. So we've started to map some of the two phage onto the tree, and so this started out as a sort of practical question, which is that if we can identify these groups in different hospitals, clusters in particular wards, and the difference in the hospital in Izmir, and indeed the difference between Turkey and the rest of the world, can we just, does it make sense to sort of pursue a kind of a crude, quick and dirty PCR assay to identify these different clades. Now we know what the clades should be. And we did this on the basis of phage sequences. And it worked pretty well, just assigning PCR primers to the different phage sequences. And then we looked a bit more closely at the phage sequences themselves. And one of the clusters, of the one in Istanbul, which I didn't have time to talk about, is actually associated has picked up a phage, even though on the core data doesn't look related to the Izmir isolates. It's actually picked up an Izmir phage. So it may be possible that in that case, that acquisition may have something to do with this epidemiological characteristics. It's quite striking. In other cases, there's no really clear story at all. They seem to be a bit all over the place. So it's a very interesting question. I don't think there's going to be a general pattern, but I think that there may be cases where phage are associated with clusters of epidemiological relevance, and that may help look, I mean, that's interesting from a biological point of view, but it's also interesting from a practical point of view in being able to identify these clusters very quickly. And in the end, we've now got a tree for the whole species. So once we can be confident for that, that we've really pulled out all the mess from the core, then we start mapping the gains and losses of all these different elements on a much longer scale. And it'd be very interesting to see, how the dynamics of those that vary between the complexes and between the different elements, and that's really something that I want to get into in the next year or two. Yeah.
I mean, presumably you can go back to the isolates and you can manipulate them and test questions about biology.
Yeah, absolutely. Yeah. No, that's right. Yeah, okay. And you had a second question about virulence, but I'm not sure that I said that I didn't mean to say they were increasing virulence. So ST239 appears to be more virulent in hospitals. I'm not sure it's increasing virulence. Is that so? I'm not quite sure what you.
Well, that was my impression that MRSA was becoming more virulent, but maybe I misread it.
No, I don't think so. No, I think the 239. There's a couple of papers which suggest that it has increased biofilm forming, etc., capacity, etc. But I don't think there's a general trend that it is. No.
But, if I can follow up, that actually gets to some very interesting aspects of the ecology of strain displacement.
Absolutely.
So, phages, as Bruce and I were discussing this morning, are a lot like bacteriocins in terms of colonization factors, lysogenic phages in terms of displacing other resident flora, which is interesting. And the other thing that Michael pointed it out in the U.S. that although MRSA was long established in hospitals, mainly confined within clonal cluster 5 type strains, USA 100, 800, that even though that was well established for quite a while, it didn't take off in the population until that went over into the sequence type 8 strains, the clonal cluster 8 strains. So and that gets to the question: virulence, colonization. What is the relative difference of these strains? Because that really is a stark transition.
Absolutely.
Seems like.
I'm very pleased to hear the word ecology being mentioned over and over again in these last 2 days, because I think it's a new.
Mark paid me to do that.
Right. Well, that's great because it's really, I think, marks a fundamental shift from looking at these things, these bugs through our anthrocentric goggles. These are the things that cause disease. It's actually thinking what it's like to be an aureus. What does aureus have to contend with? What's the role of the phage in maintaining all this, the variation that we see in the population? Why is it that the penicillin resistant strains haven't gone up to 100%? Is there some sort of balancing selection there? Why do you never see? In all the carriage studies I've never seen any particular lineage predominate more than about 35%. So there looks to be some selection maintaining diversity in the population, and I'm really, really pleased to get the impression that the ecology of the bug is really on the agenda now. So that's famous.
Other questions?
I have a question for the two network modelers here. People often cite bridges as being critical elements in containing the spread within a network. So if you have a bridging element, that might be an ideal place to intervene, between two clusters. And I didn't hear either of you mention much about that today. So if you have one hospital that's bridging between two clusters, perhaps that would be a place to intensively scrub. And I don't know if either of you have tried that in your models to see if it makes a big difference in the outcome of MRSA dissemination.
Within the English network, we see a couple of hospitals that have sort of a bridging function between between clusters, and there's actually one that connects two clusters in joining them. And if you would take that hospital, it's easy to sort of prevent spread from one side of the cluster to the other side. Yes, you can see them. Yes.
Yeah, so typically in social network analysis, there's the term broker, where an element or node actually serves as the connection between two groups or clusters. And it's definitely true in a heterogeneous network that you can see certain clusters of hospitals which are not necessarily connected to each other, with the exception of one or two hospitals that are connecting them. And these brokers can come in unusual forms and unusual locations. One of the things that we want to emphasize with the network analysis is there is some correlation geographically. You know, hospitals can be more connected to hospitals that are closer to each other, but not necessarily. So hospitals can be connected at very far distances from each other.
And there's a lot of connections between hospitals. So even if you take out one hospital, there's a lot of weaker connections that still connect parts of the network with each other. So taking out one is mostly not enough to prevent spread.
Okay, well, with that, let me make 2 points. One is, as Mark mentioned, the comments that I was going to have in the next session will not take place in favor of ceding that time to a discussion, which I think is time better invested. If you have questions, I'd be happy to discuss other things with you. Secondly, Ed, Chiba, Susan, Bruce, Michael, thank you so much for a terrific session, and we'll reconvene at when? I should know that, and I apologize for putting you on the spot. Sorry? 11:05. So back in here at 11:05 sharp. Thank you.